brain sends a signal down to the synaptic knob. It is the action potential, and then when it reaches down, it opens the voltage gated calcium channel, and it lets the calcium in, where the calcium gives a command for the ACH, acetylcholine, to go out and go to the synaptic cleft, and it is received by the ACH receptor in the sarcolemma. Action potential generated is propagated along the sarcolemma and down the T tubules, and when it reaches the T tubules, it goes to the sarcoplasmic reticulum, where the action potential triggers the calcium to get released from the terminal cisternae. And then what happens is, when it goes out, it binds with the troponin, and when it binds with the troponin changes shape, and thus removing the blocking action of the tropomyosin. Now it releases the actin active sites is exposed. The binding site is exposed, and the myosin head binds with the actin. Contraction, myosin cross bridges alternately attach to actin and detach, pulling the actin filaments toward the center of the sarcomere. Release of energy by ATP hydrolysis powers this cycling process. So once it is over, this calcium is removed by the active transport, and it goes back to the sarcoplasmic reticulum, and that ends the action potential. The tropomyosin blockage is restored, blocking actin active site, and the contraction ends and the muscle fiber relaxes. Okay, so now what happens here when it's contracting is that ATP binds the myosin, making it detach from the actin. So the ATP is the one that detaches it. And then ATP turns into ADP, releasing energy to convert myosin head into a cocked state, where it is ready to hook up again with the actin. Calcium binds the troponin, which exposes the binding site on actin that myosin can grab. And after that, the myosin now attach to the binding site, and then completes a power stroke pushing the actin. And then afterwards, it goes back to where the ATP binds the myosin to make it detach from the actin.